Today we're doing genomic technology. We did watch the video yesterday from Jimmy Kimmel about people not knowing what GMOs are. Yeah. We finished it? Okay. Where's my notes? There they are. All right. So to kind of pick up where we left off, I mentioned GMO, and as you all know, a GMO is a genetically modified organism. Spider-Man is a GMO. Captain America is a GMO. It's the American thing to do. Non-specific, oops, non-scientific term. The definition of GMO is simply any organism that has had its genome Now remember that your genome is your entire DNA, all of it. The entire genome modified in some way. As I said earlier, I can guarantee you, which is something I don't always say in this class, that you all are eating GMOs regularly. Here in Florida, if we weren't called the Sunshine State, we would be called the Citrus State. We have, in college football, you got the Citrus Bowl, you got the Orange Bowl. There used to be something called the Tangerine Bowl. Well, it's a pretty popular industry in here, Florida, citrus. In fact, during baseball season, spring training, all the teams in Florida are called the Grapefruit League. So the problem is that your traditional Florida orange is actually very, 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 very acidic, tastes gross. You wouldn't want to eat it. So we actually have blended it with other citrus plants that are more palatable to try to lower the acidity of your traditional Florida orange. So here in Florida, you have tangerines, you have tangelos, you have mandarins, you have those little cuties. And then you have the big ones like grapefruit. So there's a lot of different varieties, not just oranges. But some oranges are pretty nasty, but we've genetically modified them to be a little more palatable. Now, the next item is a transgenic organism. Trans means change. This is any organism that has a gene that has been transplanted from another organism. The other day when we were doing mutations, I was asking if anyone here is a Marvel geek. A couple of you said, yeah, I can be at times. When Thor Love and Thunder started filming this week. Oh boy, and guess what? Thor lost all his weight. He's not Fat Thor anymore. Sorry. If you like dad bod Thor, he's gone. Guardians of the Galaxy are going to be in it. Oh boy. Well, anyway, in Marvel, you got Spider-Man. A lot of Spider-Man's villains, his enemies, are transgenic organisms. You got Morbius, the living vampire. He has bat DNA inside of him. You got the lizard. Obviously, he has lizard DNA. The rhino, the scorpion. Even Spider-Man has some spider DNA in him. Duncan has some GQ DNA in him. And look at this guy. Yeah. So a transgenic organism is going to be an organism that has another organism's DNA in it. Recently, in the last couple of years, scientists have actually, just through trial and error and crazy experiments, they have found that jellyfish have a gene that can basically, I don't want to say fight against, but can curtail neurological degeneration. There's a gene in jellyfish that might be a cure for Alzheimer's. I mean, who's looking in a jellyfish? But that's what scientists do. They pursue leads, they test things out, they experiment, and if something has no promise, then they move on to the next thing. Then someone just got in their head, let's look at a jellyfish, which doesn't really even have a nervous system. And lo and behold, it can stop your nervous system from breaking down. Might be an Alzheimer's cure. Well, when it comes to transgenic organisms, one of the things you want to target from them is resistance genes. And these are mainly used for plants. So as you saw with the Monsanto story, some plants can be genetically engineered to be resistant to herbicides. Herb means plant, and side means kill or death. So you have some plants that resist herbicides. You can go to your garden and you can spray your herbicide and you don't have to worry about killing your tomato plants, killing your wheat plants or your corn. You're only going to kill the weeds. But here's something that's actually happened is when you have these genetically modified crops, somehow the gene that resists herbicide is able to transform, which means just move, into the weeds. And so the weeds get to be herbicide resistant too. So that has happened. Another is going to be resist insects. There are some plants that have built-in insecticides because it's not really a promising business venture to constantly spray your food with bug killer because beetles and caterpillars will love to eat your crops. So what scientists have been able to do is to genetically engineer plants to have a built-in pesticide. So when a caterpillar or a beetle tries to eat it, it is really, really gross and they don't want to eat it anymore. It's not going to hurt humans. There's no record of it hurting humans. But it's a lot better than just like spraying bug killer all over your crop. And here's another one that's really popular up north, resist freezing. Here in Florida, when it gets really, really cold, maybe 2 or 3 nights a year, you'll hear about in Plant City, the farmers of the strawberries will spray their crops with water. And so if we're in a freeze warning, which inland they were in a freeze warning, it got below 32 degrees Fahrenheit. To prevent the strawberries from dying from the cold, which is called like cold shock or freeze shock, you coat them in a layer of water, and that water freezes, and it actually protects the strawberries. So every year, when we have that 1 or 2 nights a year where it's going to get below freezing down here, you'll hear the farmers in Plant City are spraying their strawberries so they can form a layer of ice around the strawberry, and they'll be protected. Well, up north, when it freezes a lot, you could kill the plant. The plant's going to get frostbite. And so they'll insert a gene that resists freezing. These plants have antifreeze. It's crazy stuff. Now, all these GMOs are really important, and the majority of people on Earth eat them because when we get to Unit 8, which is ecology, you guys are going to learn about something called a carrying capacity. Earth is not designed to be able to handle 8 billion humans. It's just not happening. So that's where we have to actually use our technology and our advancements to try to cheat the system. And one of the things we can do is use our technology to make as much food as possible. If you were to take all the GMOs and all the technologically created food out of the picture, billions of people would die. Billions of people. And whoever's left is going to be having war over whatever food is left. And so our society, not just American society, but our human society is dependent on these GMOs and genetically modified organisms and transgenic animals and plants. Now, when it comes to both plants and animals, you can have things called growth factors. Yesterday, I showed you an image of this really, really, really big cow. This is like the Dwayne Johnson of cows. It has a lot of muscle. So these organisms, and it's in plants and animals, it's not just plants and it's not just animals. They make organisms grow faster and produce a greater yield. So if you had a cow that when you took the cow to slaughter, it made, let's say, 80 pounds of meat. Well, if you were able to engineer a bigger cow and let's say get 150 pounds of meat, money. And that's what they do. They make these cows bigger to have more meat. They make turkeys bigger, ducks bigger, pigs bigger. Heck, if you go to Publix, you go in the produce section, they have these things called beefsteak tomatoes, which are half the size of your head. They're huge. Yeah. No, these cows are ripped. There's like not an ounce of fat on them.
Because I remember watching this like documentary once, and it was like the chickens, like their legs, like they can't move because they're so fat, and like, that makes the meat like more like fatty.
Well, I guess that could be a problem. That's where you have to just refine the science, and you want to work more with the muscle. I will tell you that here, y'all don't need to write this down, but for those of you that do remember from Unit 1, what is the key element in protein? Oh boy, you're freaking me out. Andrew, you look like you want to raise your hand, but you don't have the guts. Jasmine? That's the monomer. I'm talking about element from the periodic table. Ryan? No, everything is carbon. Yeah, it is nitrogen. So nitrogen is in the compound called ammonia. So what a lot of farmers do is that they will spray ammonia all over the hay that cows eat. And so the cows are getting extra nitrogen, which means they're getting basically extra ingredients for muscles. And so the cows will actually get bigger, more muscle. What? It's not concentrated. They're not like chugging a bottle of ammonia.
Is that where there's like a lot of nitrogen and protein powder and stuff like that? Like N3.
Yep. All right. Now, here is where we're going to really get into some interesting stuff. Here, how does it affect human health? Medical, transgenic organisms. These are going to be organisms that produce important medicines due to gene insertion. And I'm going to definitely talk about two, and if we have time, a third example of this. This is probably the most groundbreaking technology that we have outside of things like vaccines. So the first thing that I want to discuss when it comes to this topic is treating genetic diseases. And the genetic disease that I would like to focus on is sickle cell anemia. So the first example I want to use when talking about medical and transgenic organisms is treatment for sickle cell anemia. And to save time, I'm going to just abbreviate sickle cell anemia, SCA, sickle cell anemia. Now, a couple months ago, we watched a video of this young African-American girl that she was like 24 or so. She had sickle cell anemia, and it's actually most common in people of African descent because that's where the mutation evolved from, Southeast Africa. It's a defense against malaria, but it also can cause excruciating pain because the blood cells turn into a hook shape, which clog up your blood vessels, and it causes things like blood clots. You saw that when at the end of the story where she was actually more or less cured. She was doing karate, she was jogging, she was swimming. Any one of those things would have likely killed her if she still had the disease. So a genetic disease is woven into your genome. It is part of who you are. Just like Ryan has brown hair, that's part of who he is. I'm tall, that's part of who I am. Well, these genetic diseases are part of who these people are. And it was really thought you really can't cure a genetic disease, but recent technology has actually shown, yeah, you kind of can. And so when we were watching the story, The doctors who treated her said that they used HIV. And here's what they did. So before I show you what they did, I actually need to show you how HIV works and why they used HIV. So let's talk about for about 5 minutes about what HIV is. So in treating sickle cell anemia, they used the virus HIV. HIV is the human immunodeficiency virus. It's a virus that attacks your immune system and basically leaves you without an immune system. So you die of things like pneumonia, the cold, the flu, stuff that you normally would be able to fight if you had a fully functioning immune system. So HIV is known as a retrovirus. I think COVID's a retrovirus. I'm not sure. Well, a retrovirus has mRNA instead of DNA. Some viruses have DNA. Some viruses have RNA. They typically don't have both. Well, here's the problem, guys. A virus is not a living thing. Let's be clear about that. If you ever hear someone call it an organism, wrong. It's called an obligate parasite. It does not have its own branch on the tree of life. Instead, it's kind of like the vines on the tree of life. It's been evolving with us, and it knows how we are put together. That's how it can infect us so easily. Well, a retrovirus is any other virus. It needs a host to survive, and it really needs a host to replicate itself. That's the goal. And so what these viruses do is that they first have to hack into your nucleus. So let's say here is an HIV virus, and inside the HIV virus is some viral mRNA, which is a single-strand nucleic acid. So the first thing that this virus has to do is it has to get into the nucleus of your cells. Now I'm going to tell you why it has to do that in a moment. But there is a process called reverse transcription. Duncan, when you have regular transcription, you go from what to what? Yeah, in normal transcription, you go from DNA to RNA. So Duncan, don't overthink it. What do you think the order is when you do reverse transcription? You start with RNA and you make DNA. So in reverse transcription, you go from mRNA to DNA. There's an enzyme that lets you do this. Remember that enzymes are named after what they do. It is called reverse transcriptase. How do you know that that's an enzyme? It ends with the letters ASE. This is the enzyme that actually makes reverse transcription happen. Okay, so over here, I'm going to draw a host's DNA, like a human. Maybe it's Jenny from Forrest Gump or Eazy-E from Straight Outta Compton, both people of whom got HIV. So this is host DNA. All right? Well, the virus has gotten into your nucleus, and it is going to perform reverse transcription. So that you will have sections of the host DNA that has the virus. So as you all can see here, we now have viral DNA, and that's big. So this is what the virus has accomplished. It has used your nucleus to make viral DNA when it didn't have DNA before. So why is it trying to get to your DNA? And why is it trying to make DNA? Because as Duncan said, when you go from DNA to RNA, you have transcription, and you can make a whole lot of RNA. So when the viral DNA goes through transcription, it makes a lot more viral mRNA. And all this is, what this basically is new viral mRNA, which results in new viruses. They're kind of like quote unquote baby viruses. And all these new quote baby viruses will go into your other cells and repeat this life cycle over and over and over. That's how HIV works. And it just so happens that the cells that it targets are your immune cells. Hold on. Now, some viruses will actually stay with you your entire life. When scientists conducted the Human Genome Project, they learned that about 8% of the human genome is ancient viruses that our ancestors experienced for thousands of years. Have any of you ever gotten a cold sore, like on your lip? I'll say I have. That's called herpes simplex 1. Now, herpes simplex 2 is the STD. Well, herpes is a virus. You ever see The Hangover? Well, in The Hangover, the beginning of the movie where the future son-in-law, Doug, who ends up getting lost, his fiance's father says, here, take my car, enjoy yourselves. And Doug is saying, are you kidding? Oh my gosh, you're so nice, thank you. And the father goes, now remember, what happens in Vegas stays in Vegas, except herpes. That'll come back. Herpes stays with you forever. When I was a child, I would get the herpes simplex 1, this little cold sore on my lip, like every year around February, now. And my cousin was a dermatologist. So my dad took me to the dermatologist and he gave me this medicine that made it go away. And I didn't have it for 14 years. And then I was 24 years old driving home from work one day and I looked in the rearview mirror because I felt something weird on my lip. I'm like, oh no, it's back. 14 years later. So it stays with you forever. And I will maybe have it pop up once every 3 years. It sucks, but I think I had it a few months ago and it's gone. So hopefully not for a few more years. But when I'm an old man, I still have I will still have this virus in my DNA. It's there forever. Some viruses do go away, some don't. Well, now let me show you how we actually use HIV for treatment in gene therapy. Yes, Ayla, you had your hand up.
What like dictates whether it comes back or goes away?
That's a good question. So how does it come back? It's your immune system gets weakened somehow. One of the things that can weaken your immune system is UV radiation. So I remember when I was 24 and I had the little I had the little herpes simplex 1 sore come back. I was at the beach that weekend before. And so when you get a lot of UV radiation, that kind of weakens your immune system temporarily. And it's weak enough for the virus to kind of make a little cameo appearance.
And for COVID, is that why there's like so many kind of like different symptoms or like
They don't know why there's so many symptoms. People that have been epidemiologists and virologists their whole adult lives say that they have never seen a virus that has such a wide range of symptoms like COVID. They don't know why. All right. So let me show you guys now what happens. So check this out. First off, you have HIV, but you have removed the viral RNA. So think of this analogy. Let's see how this sounds. You have a horrible man driving a car. You take out the horrible man. The car's fine. A crack house is only a crack house if there's drug dealers in it. You take out the drug dealers, it's just a house. If you have a virus that does deadly things, you take out the viral instructions, it's just a container. And so what they put inside instead is non-mutated hemoglobin gene. This is what actually can cure people with sickle cell. People with sickle cell have a mutated hemoglobin gene. Well, thanks to technology and science, scientists have learned how the correct gene should look. And so they'll build it in a lab, they'll put it into a virus, and now this is like a car being driven by a good driver. And so we now have the host DNA, like that young woman in the video we watched. Or I guess it's not really the host anymore, she's the patient. And you're going to have reverse transcription. And now, over time, oh, by the way, if you could label this DNA as being mutated, remember, this DNA has the sickle cell anemia mutation. Well, now it's non-mutated. This gets implanted in, or this gets put into stem cells, which are put into a patient's bone marrow. Bone marrow is where your red blood cells really come from. And so if you have a mutation in your blood cells, there's probably a mutation in your bone marrow. You need to fix the genes in your bone marrow. So you are basically getting infected, but this is a good infection. It's infecting you with non-mutated DNA, and it's going to change your DNA from being mutated to non-mutated. So it is kind of like I said, getting infected, but in a good sense. The virus is doing what it does, but it's carrying hugs and kisses instead of bullets and knives to hurt you. And this is how that woman was cured. And if we can do that with sickle cell anemia, we could do it with Tay-Sachs, we can do it with Huntington's, maybe red-green color blindness, hemophilia, cystic fibrosis. The opportunities are endless. All right, now, the other example is going to be recombinant DNA. And this is what your activity on Tuesday is going to be about. For this science, we use the prokaryote E. coli. E. coli is the microorganism that lives in your intestines. It is used to treat diabetics. When you have diabetes, you're type 1 diabetes to be exact. Type 1 diabetes is an autoimmune disorder. Do any of you know what autoimmune means? What does auto mean? Self. In an autoimmune disorder, your own immune system is attacking yourself like it thinks that you're the bad guy. If you've ever heard of multiple sclerosis, your immune system is attacking your neurons. If you have rheumatoid arthritis, your immune system is attacking your joints. If you have Crohn's disease, your immune system is attacking your intestines. Well, in type 1 diabetes, your immune system is attacking the beta cells of your pancreas. That's where insulin just so happens to come from. So if you have type 1 diabetes, your pancreas can't produce insulin because your pancreas is under attack. Your insulin is needed to escort sugar that you eat into your cells so that your cells can do cell respiration. When you have type 1 diabetes, you can't do that, so you have to take an insulin shot. Well, the way that this used to go is the insulin would come from pigs. We would give pigs oodles and oodles of sugar, sugar water, sugar food, sugar slop, everything. And these pigs would be so fat and out of shape and diabetic, and they would eventually die. But we did all that to try to get as much insulin from them as possible, and then we would package up that insulin to diabetic humans. But then we had the technology to do this procedure, and I can tell you that no pigs are harmed. It is far cheaper and way more efficient. You make tons of insulin by this method. So what we do is we insert the human insulin gene into an E. coli, which is a bacteria, plasmid. Now, I haven't really talked about what a plasmid is, so I'm going to tell you now. Let's say that this is an E. coli cell. It's a single-celled organism. And inside you have the little cloudy area of DNA. That's called the nucleoid. Well, just to the outside of the nucleoid is a ring of extra DNA called a plasmid. And again, this is just extra DNA. This is what scientists use to insert the human gene into the bacteria. So we're going to take that plasmid, and I'm going to draw it like this. There it is. Kind of zoomed in. And we're going to focus in on this area right here, okay? So let's zoom in on that section. And we're going to zoom in on this part where it goes, let's see, GAA, TTC, which means the complement is CTTAAG. This gene sequence is called the restriction site. This is where you're going to open it up. Yesterday, we were doing gel electrophoresis, and I introduced the enzyme to you called a restriction enzyme. A restriction enzyme is used here. It is going to open up the gene. Where an A and A and G are. So wherever you see a G and an A side by side, that is going to be where this genetic code opens up. So if you look at this, do you see a G and an A next to each other? Yeah. So here it is. Watch. Let's say that you were using scissors. You're going to cut along that line. And so the opened up gene will look like this. Okay. These ends here are called sticky ends. This is where you're actually going to plug in, for lack of a better word, or splice the human insulin gene. That's where you're going to put it, in there. So the human insulin gene will look like this. All right, now I'm running out of time here, guys, so I'm going to try to improvise with this. I want to show you what I'm going to do. So if you can just pay attention, I don't want you to miss this because you're looking up and down at your notes and you're looking at the screen. So watch this. I'm just going to move this away a little bit so I have some space. I'm going to erase this, and here's where the magic happens. We're going to insert this human gene, which I've drawn in red. This is the human insulin gene. I'm going to lasso that. I'm going to splice it in here. I know it's not perfect fit, but trust me, it'll fit right there. And you have now inserted a human DNA sequence into a bacterial genome. This is known as recombinant DNA. And if you actually zoom out, it's going to look like this. And this bacteria, this E. coli, is going to make a tremendous amount of insulin for pharmaceutical companies. And they will sell it to diabetics because the diabetics need that insulin in order to metabolize sugar. This is what you're going to do on Tuesday with a simulated bacterial plasmid and a human DNA sequence. Okay? All right. That's all I have for you today. You will see this again on Tuesday, so now you hopefully have a bit of a foundation.